All right, let's jump right into this explainer. We're tracking the incredible journey of a post-mortem genetic sample, literally taking it from complex molecular science all the way to the courtroom. It's a true multidisciplinary puzzle. I mean, have you ever wondered why two different people can take the exact same dose of a drug and one walks away totally fine while the other suffers a fatal toxicity? Well, the answer is often hidden right there in their DNA. Today, we're unpacking exactly how that works and how forensic toxicology is using this step-by-step science to solve some seriously complex cases.
So here is our roadmap for today. We'll start with Step 1, the pharmacogenetics mechanism. Then, Step 2, optimizing the lab workflow. Step 3 is navigating post-mortem consent, and we'll wrap up with Step 4, legal admissibility in the court. It's a complete guide from start to finish.
Kicking things off with Section 1, the pharmacogenetics mechanism. Before we get into the lab or the law, we absolutely have to get our foundational science straight.
For any undergraduate forensic science students joining us, think of pharmacogenetics as decoding the body's unique instruction manual for breaking down chemicals. Officially, it's the study of genetic variations influencing drug pharmacokinetics or pharmacodynamics. But in plain English, it's basically the science of how your specific DNA sequence dictates how your body absorbs, metabolizes, and eliminates foreign substances, like opioids or antidepressants.
Let's look at how we actually categorize these variations. Take the CYP2D6 gene, for example. It's a major player in the cytochrome P450 enzyme family and handles metabolizing drugs like codeine, morphine, and methadone. Think of it like a highway. Some drugs speed through the system. That's your ultra-rapid metabolizer in the fast lane. Extensive metabolizers, they're cruising at the normal speed limit. Intermediate metabolizers are driving with the brakes partially pressed down because of reduced enzyme activity. And poor metabolizers, they're stuck in a massive traffic jam, which unfortunately leads to a toxic pileup.
And here's why this is absolutely crucial. Studies show that 7% of individuals who succumbed to opioid overdoses actually exhibited that ultra-rapid phenotype. It actively intensified the toxic effects. Just let that sink in for a second. For these folks, their own genetic code turned a standard, prescribed dose into a fatal one. It just proves that medical examiners can't solely rely on raw toxicology numbers anymore. They have to account for individual gene variations to find the true cause of death.
Moving right along to Section 2, optimizing the lab workflow. Because now we have to talk about the practical how, managing these postmortem samples in what is often a super busy, resource-limited forensic lab. When you're dealing with a high workload environment, implementing a practical, cost-effective workflow is just essential. You have to be smart about it. Step 1 is collecting the post-mortem samples. So we're talking blood, hair, saliva, or even spleen tissue. Step 2 is getting them into optimal storage. Step 3 is utilizing batch processing for DNA extraction. Batching is a massive time saver when you're swimming in autopsy workloads. And finally, Step 4 is running the actual toxicological investigation using precise equipment like GCMS or LCMS. Now, a really crucial part of this workflow is Step 2, storage. Practitioners have to carefully compare DNA recovery efficiency over time, specifically looking at EDTA tubes versus sodium fluoride and potassium oxalate storage tubes. To make a molecular autopsy successful, you need consistent, high-yield DNA extraction. Finding the exact right anticoagulant tube is really the foundation of that consistency for any busy lab. Okay, Section 3, navigating post-mortem consent. This is where we shift to the ethical side of things, because science doesn't just happen in a vacuum. It involves real families who are dealing with unimaginable loss.
So the big question is, how do we justify the risk versus benefit ratio for post-mortem genetic studies? Before rolling this out in forensic institutions, we have to objectively weigh the scientific benefits against the ethical risks to the relatives. Our pursuit of scientific truth can never outweigh our compassion for the families left behind.
According to research out of Cape Town, South Africa, treating grieving families as a highly vulnerable population is an absolute ethical necessity. Legally speaking, next of kin aren't always formally recognized as a vulnerable group, but come on, the shock of a sudden, unexpected death means we absolutely have to treat them as such. Maintaining profound empathy and total clarity during the consent process is just critical. We also have to consider that genetic testing can uncover really unexpected and distressing incidental findings. Think about things like previously unknown paternity issues. Because of this, strict ethical protocols are non-negotiable. Remember, you aren't just testing for drug metabolism, you're mapping out a family's genetic history.
Researchers need clear funding to validate these findings, and the results have to be delivered exclusively by a trained genetic counselor to make sure the family is fully supported. If we put ourselves in the shoes of that genetic counselor for a minute, it's all about using simple, empathetic analogies. We might say to a grieving parent, think of your loved one's body like a specific type of engine. It simply processes this fuel differently than most. Our goal is to help you understand their unique genetic pathways without assigning any blame for the toxicology results. It's so vital to remove any sense of blame and make it perfectly clear that it was just their biology responding differently.
Which brings us to our final stop, Section 4, Legal Admissibility and Court. This is where we evaluate how all this science actually holds up in front of a judge.
So let's adopt the lens of a legal advisor or a forensic expert who has to justify this testing. We have to look strictly at evidentiary standards. Because honestly, it doesn't matter how groundbreaking your pharmacogenetic mechanism is if the judge refuses to let the jury even hear about it. In court, the judge acts as a gatekeeper, and the genetic evidence has to impartially meet the criteria outlined by the Daubert standard. Think about it from the bench's perspective. Has the basic theory been tested? Has it been peer-reviewed? Do we know the potential error rate? Is it generally accepted by the scientific community? And finally, are there strict standards controlling the lab techniques?
The bottom line is that a forensic toxicologist can't just extrapolate wildly from an accepted premise. They have to clearly demonstrate how the data meets those exact court requirements. Thankfully, mechanisms like CYP2D6 are heavily peer-reviewed, and lab techniques like GC-MS and PCR have well-established standards and error rates. But the expert witness must always account for alternative explanations for the drug toxicity to survive a tough cross-examination.
All of this leaves us with a pretty provocative thought. As molecular biology continues to blur the lines of traditional toxicology, are we looking at the new standard for uncovering the truth? Will pharmacogenetics transition from this specialized tool into a mandatory, routine part of every single sudden death investigation? Understanding how our DNA, lab workflows, ethics, and the law all intersect is really just the beginning. Thank you so much for joining me as we decoded this explainer.